What if I told you that the bubbling jar of sourdough starter sitting on your kitchen counter could be the key to unlocking your garden's true potential? For years, we've been searching for ways to build healthier soil, spending money on expensive amendments and commercial inoculants, when one of nature's most powerful fermentation cultures might have been right in front of us the whole time. Today, I'm taking you on a journey into one of the most unconventional experiments I've conducted as a gardener, exploring whether the same living cultures that make our bread rise could revolutionize how we think about soil health. This isn't just theory or wishful thinking. This is a hands-on investigation into whether fermentation is the missing link in building truly living soil. Before we jump into the experiment itself, we need to understand what we're actually working with when we talk about sourdough starter. Most gardeners think of sourdough as simply a bread-making ingredient, but it's far more than that. A sourdough starter is essentially a living ecosystem, a symbiotic culture of wild yeast and beneficial bacteria that work together to ferment flour and water into something magical. The primary organisms in an active sourdough culture are Saccharomyces cerevisiae, the yeast, and Lactobacillus, the bacteria, along with numerous other microorganisms that develop over time depending on your environment and feeding practices. The real revelation is recognizing the parallel between what sourdough does in bread and what beneficial microorganisms do in soil. Both ecosystems thrive on biological activity, diverse microbial communities, and fermentation processes that break down complex compounds into simpler, more available forms. When bacteria ferment, they're essentially pre-digesting organic matter, making nutrients more bioavailable, exactly what we want happening in our garden beds. The wild yeasts and bacteria in sourdough starter have evolved over months or years to create an incredibly stable, productive culture. These aren't lab-created microbes, they're organisms that have proven their resilience and effectiveness in a real-world environment. So the question becomes, could we harness this same biological power to create healthier soil? The logic is compelling. Garden soil needs diverse microbial populations to cycle nutrients, suppress diseases, and promote plant health. Commercial soil inoculants are expensive and often contain limited species, but a robust sourdough starter contains multiple species of beneficial microorganisms that are already proven to work together synergistically. If we could introduce these cultures to our soil in a format that keeps them alive and thriving, we might be creating a self-sustaining biological amendment that continuously works for us. To truly understand whether sourdough starter could work as a soil inoculant, we need to examine the specific microorganisms at work and how they might benefit soil health. A typical mature sourdough starter is dominated by lactic acid bacteria, primarily Lactobacillus plantarum and Lactobacillus brevis, along with wild yeasts. These bacteria produce lactic acid as a byproduct of fermentation, which lowers pH and creates an environment where beneficial microbes thrive while pathogenic organisms struggle. This is crucial for soil health because disease-causing fungi and bacteria often prefer neutral to slightly alkaline conditions. The yeasts in sourdough, particularly Saccharomyces cerevisiae and other wild yeast species, produce enzymes that break down complex polysaccharides and proteins. When these organisms work in soil, they could theoretically enhance the breakdown of organic matter, making nutrients more available to plant roots. Additionally, yeasts produce B vitamins and other compounds that support plant growth and beneficial soil microbes. The fermentation process itself generates organic acids and other compounds that can improve soil structure and nutrient availability. What's particularly exciting is the concept of competitive exclusion. When you introduce a strong population of beneficial microorganisms to soil, they occupy ecological niches and consume resources that pathogenic organisms might otherwise exploit. The diverse bacterial population in sourdough starter means we're introducing multiple species with different metabolic capabilities, which translates to broader functional benefits in the soil. Unlike monoculture inoculants that contain only one or two species, sourdough brings a community, a microbial society that has already learned to cooperate and function together effectively. To properly test whether sourdough starter could function as an effective soil inoculant, I designed a controlled experiment that would eliminate variables and give us clear, comparable results. I selected 4 identical tomato plants of the same variety and size, all at the same growth stage. I divided my garden space into 4 distinct beds with identical soil composition, the baseline soil being moderately healthy but not biologically supercharged. This was crucial because I wanted to see if sourdough starter could meaningfully improve upon already decent soil. For the control group, 1 plant received no amendment whatsoever. The second plant received a conventional commercial soil inoculant according to package directions, representing the standard approach most gardeners take. The third plant received compost tea, which is a well-established fermented amendment that many organic gardeners swear by. And the fourth plant, our wild card, received sourdough starter inoculant that I'd prepared specifically for this experiment. The sourdough preparation was key. I took an active, healthy starter that had been fed regularly and maintained at peak activity for several months. I diluted it with non-chlorinated water in a 1:5 ratio and allowed this mixture to ferment for an additional week, feeding it with a mixture of flour and water to keep the culture thriving. This extended fermentation period allowed the microbial populations to multiply significantly while remaining in active, viable form. I applied this preparation directly to the soil around each plant at planting time and again at the 2-week mark, following similar application rates to the other amendments. The initial setup involved daily observations to monitor plant emergence, early growth patterns, and overall vigor. I measured soil temperature, moisture retention, and took soil samples to establish baseline microbial populations. I also noted any changes in soil appearance, color, texture, crumb structure, and smell, which can all indicate biological activity. The first weeks of the experiment showed interesting initial patterns. The control plant developed normally but showed the characteristics you'd expect, steady but unremarkable growth. The commercial inoculant application showed slightly accelerated early growth, though this effect appeared to plateau after about 3 weeks. The compost tea application produced notably vigorous plants with darker green foliage and more robust stem development from early on, but the sourdough starter plant was where things got genuinely fascinating. By week 3, this plant was displaying characteristics more vigorous than the control and nearly matching the compost tea plant. The foliage was noticeably darker and more developed. What was particularly striking, however, wasn't just above-ground growth. It was what was happening in the soil. When I extracted the plant at the 4-week mark to examine root development, the sourdough-treated soil showed remarkable characteristics. The soil structure was noticeably improved, with better aggregation and crumb formation. The roots had developed a more extensive root network, with visible increased branching compared to the control. By week 6, the sourdough starter plant had actually surpassed the compost tea plant in overall vigor. The foliage was lusher, the plant was flowering earlier, and when I measured chlorophyll content using a simple handheld meter, the sourdough plant registered higher values than all other treatments. The smell of the sourdough amended soil had also changed. It had a distinctly earthy, alive quality that intensified over time, suggesting robust microbial activity. One of the most revealing measurements came from soil testing. I sent samples to a lab for microbial count analysis at weeks 4 and 8. The sourdough-treated soil showed the highest microbial biomass of all treatments by week 8, with bacterial populations increasing dramatically and fungal populations showing healthy diversity. This suggested the sourdough starter wasn't just providing a one-time boost, but actually establishing and proliferating within the soil ecosystem. What emerged from this experiment went beyond simple plant growth metrics. The sourdough starter seemed to create a cascade effect in the soil, initial microbial inoculation leading to broader biological awakening. The improvement in soil structure was particularly notable, because this suggested the microbes were actively producing compounds that bind soil particles, improving water retention and root penetration. However, the experiment also revealed some important caveats. The effectiveness seemed highly dependent on the quality and activity level of the sourdough starter used. I tested a second starter that had been neglected and inconsistently fed, and it produced much less dramatic results. This taught me that not all sourdough starters are created equal. The vigor of your culture directly impacts its effectiveness as a soil amendment. Additionally, the initial application timing seemed to matter significantly. Applications made during active microbial growth phases appeared more effective than those made with overgrown or underfed starters. The implications of these findings open entirely new doors for gardeners interested in building living soil. If a sourdough starter can establish beneficial microbial communities in garden soil, it means gardeners can create their own custom soil inoculants at home for pennies. Instead of buying expensive commercial products, you could maintain a sourdough starter specifically for your garden, breeding populations of beneficial microbes tailored to your local environment. Over generations of feeding and maintenance, your garden starter would develop local microbial populations adapted to your specific climate and soil conditions. Future experiments worth pursuing include comparing different types of fermentation cultures, wild fermented vegetables, kombucha starter, or kefir, to see if other fermentation systems produce similar results. Testing sourdough starter on different plant types could reveal whether the benefits are universal or specific to certain crops. Long-term soil health tracking could determine whether sourdough inoculation produces lasting improvements or requires repeated applications. This experiment reveals something profound about gardening in the modern era. Sometimes the most powerful solutions hide in plain sight, waiting for someone to ask the right question. Your sourdough starter might genuinely be one of the most valuable biological tools in your gardening arsenal, and it costs almost nothing to create and maintain. The fermentation cultures thriving in that kitchen jar have spent months or years evolving to be resilient, productive, and beneficial. Why not harness that power for your garden? I encourage you to conduct your own version of this experiment. Maintain an active sourdough culture specifically for garden use if you already have one, or start one today. Document your observations, compare treated and untreated plants, and measure the changes in your soil over time. This is citizen science at its finest. Real gardeners testing real methods and building collective knowledge about how to grow better plants through truly living soil. Subscribe to the Living Soil Lab to stay updated on ongoing fermentation experiments, soil biology discoveries, and unconventional growing methods that challenge conventional wisdom. Share your own sourdough soil experiment results in the comments. I want to hear whether this works as spectacularly in your garden as it did in mine. Together, we're building a community of gardeners who understand that the future of productive agriculture isn't found in bottles and bags, but in the incredible biological potential of fermented cultures and living soil. The revolution is happening in kitchens and gardens everywhere, and it's time you join the movement.